Hey, everybody, let's find a seat so we can get going, or else you all have to stay until 6 o'clock. No, not really. I'm Joyce Suji from Exponent. I'm on the Emerging Sciences Health Decision-Making Committee. And I'll be announcing the next group of speakers, so please stop your conversations. I know it's all really interesting, but let's get through this. Our next speaker is James Kaput with Nestle Institute of Health Sciences. Thank you.
Thank you very much, and thanks for the invite. I guess I'm the outsider here because I'm going to talk about stuff we haven't heard about today. But that's okay, I think, because it might add a little bit of thought or some different ways of thinking about this process of individuality. I want to say 2 words. I don't have any slides. I want to say 2 words about the Nestle Institute of Health Sciences. That's a separate company that was established by Nestle with a 500 million Swiss franc investment per year for the next 10 years. And its mission is to understand and study health, in particular health, maintenance, and disease prevention. So it's actually one of the few, if not the only, such institute and large commitment of its kind that I'm aware of it at any rate, because in most cases we're talking about disease prevention maybe, or disease, and all the markers that we have for disease are used to define health. That is, you have the absence of a marker for disease, and that's how we define health. In reality, we don't know how to define health, and so that's the mission of that institute. This also is a part of a 2 billion Swiss franc investment in the space between food and pharma. And I think that in the coming years, the U.S. regulatory agencies, the European regulatory agencies, are going to see a lot of products from this space in terms of regulation, and they will be for either individuals or for groups, as I'll try to tell you today. So this is what you'll see today, which is how do we do science, much of what we heard about today. What's the 20th century design that we use? What about diets, physiology, and genetics, which make it very difficult to use that design? And then I'm going to tell you how we go about looking at health processes and health research. So regardless of whether we talked about it, you all know this, particularly the statisticians. What we did in the 20th century was essentially say, I want to have someone who is normal weight versus obese, non-diabetic versus diabetic, intervention versus non-intervention with a food or a toxin. And then we put those people into some type of weighing measurements. It could be a scale for weight. It could be a mass spec for blood, genetics, what have you. But when you think about the results of that analysis, what we're getting out is the population attributable risk. That is the group average. And when you take the group average and you put them in all your models, the variability that we're so interested in gets lost. I've never been in one of these studies, so my genetic makeup nor my environmental exposures have ever been measured, and yet the risk and benefit factors for both the foods and the toxins are supposed to apply to me. And I claim that that's not true. So what we look at is, more graphically, is this distribution of health, distribution of response in the normal population, the distribution in the disease or affected population, the exposed population. Most of the time, these overlap, and we have some population attributable risk. And that population attributable risk is supposed to apply to the individual. And in fact, we use that population attributable risk, individual risk factors for, let's say, a gene, and we usually use the additive model and add up all the risk factors, and we find from our GWAS friends that for glucose metabolism, you can only explain 1.5% of the total phenotype from all of the 30 to 35 individual SNPs or loci that they've identified. And I claim that the reason that they haven't identified is they forgot the environment. And it's very difficult to measure the environment because food compositions vary depending on where you grow the food, how you process it, how you cook it. Our cultures matter around the world. And so we get exposed to different compositions of food. Now, we actually haven't discussed today yet food or nutrients, but there are bioactives in food, which alter the expression of the genes that are, that are involved in the metabolism of both the toxins and drugs and the nutrients. So you can have chemicals in food, fatty acids, sterol esters, which bind transcription factors, which alter the expression of phase 1, phase 2 genes, as well as cytochrome C peroxidase genes and esterases, all the types of genes that we know about. And yet when we measure toxicology effects or drug effects, we very rarely, if ever, measure our nutritional environment. And I think this is a very big hole in the analysis of the data and therefore what we use in our model. So I won't belabor that. I'm going to give you an example about health and disease variability, which we have not been measuring, and also this genetic variability, which you've heard about today earlier. So I use type 2 diabetes as an example. In the United States, in the old days, if you had high glucose levels repeatedly, you were diagnosed with impaired glucose tolerance, and you would be told to try diet and exercise. And let's assume that we told that to 100 people, and all 100 actually changed their diet and exercise. It turns out that about 85% or 85 out of those 100 would still require some type of drug medication in order to control their glucose levels. So we give them a variety of different drugs. The first line is sulfonylurea or biguanide. But I want you to focus on the last column. The last column shows the effectiveness of that drug. And all of us who were in the FDA or associated with drug studies know that drugs are not effective in every individual. But if you have to think about the biology behind that, why is that the case? If somebody has high glucose, how come sulfonylurea, which blocks the potassium channel, not influenced by that drug? Only 50% are. So here's some of that variability that we were talking about, or biguanide. And the reason is, I've color-coded, these two drugs work in the pancreas, or on the pancreas, to stimulate insulin secretion. So individuals who have high glucose because they have a poorly functioning pancreas are going to be helped by these two drugs. But those who have overexpression or increased flux through the gluconeogenesis pathway, which also results in high glucose in the blood, are going to be mostly helped by biguanide or metformin. And then these other two drugs work in intestines for uptake of glucose, and the TZDs in the peripheral tissue for insulin sensitivity. So type 2 diabetes is not one disease. It's a symptom of high glucose in which multiple organs and multiple pathways can be influenced. But when our colleagues in the GWAS studies did the GWAS, they lumped everybody who had high glucose together, including in one study individuals, 20% of whom did not have an overnight fast and just happened to walk into the clinic. Somehow that got into the Science magazine. In any event, only the GWAS people can get that in. I couldn't get that in. What they did is they put everything together for multiple physiological processes, and then they looked at the genes that are associated with that high glucose level, but it could be distributed amongst all these different pathways. And so that's one explanation why they identified so few genes involved in the GWAS pathway. But there's another issue that also is, I think, even more important. So on your left is the genetic map of Europe. And each individual there, each symbol, which you can't read because it's always fuzzy, is, you can think of it as the country code for the internet. So IT are Italians, and ES are Spaniards, and PT are Portuguese. My ancestors are from Poland, so it's PL. And with these type of data, they can position my grandparents' place of birth within 400 kilometers at 95% accuracy because of my, and they would study me and not my grandparents. So each individual is genetically different. And we now have maps like this for Africa, China, India, some of the smaller countries around the world. The one we're missing, quite frankly, is South America. A team that I have the reference for on the right, this map, each individual here is a circle. So they took data like this, but there's a different study. Each individual is a circle, and they tried to find the best overall genetic match in 2,457 people. And you can see there's a cluster here. It turns out there are 187 people in that cluster. And you could say that that could be a good match to do a case control study. But all the other ones are sort of separate in their own separate clusters. So if you try to do a case control and divide that population by a variety of different methods, there's no way that you can get an exact genetic match between the cases and control. In fact, it's even worse than that because there's no two-dimensional information there. So all I have to do is look at the population slightly differently, and my case control goes out the window. Now, here's a problem that I have, because these are data which are widely known throughout the genetic community, widely available to all of us. And given this genetic heterogeneity, we still design case control studies. Now, I would also argue that if you took our dietary intakes and our lifestyle, how active we are, you could probably draw a very similar map. All you have to do is watch what we had for lunch or when we go to dinner tonight around the table, and you'll know that every individual eats differently and every individual has differences in environment and lifestyle. And yet we still claim that we can find a way that we can put a case control study. And you heard about the 1,000 Genome Project. It's likely that we even have more variability than what's on this map. So here's the logic of the 20th century, which unfortunately is seeping in to the 21st century. They go, the reason we couldn't get good numbers, good p-values, and we're having all the missing heritability is because the power of our experiment was too low. We only had 1,000 people or 2,000 people in this study. So what we need are 19,000 more, or 2,000, 200,000 more. In fact, there are some calls to do a 200,000 GWAS study in the United States. But just from what I told you about the food cultures and heterogeneity, disease variability, and the genetic variability, what's the assumption that is being made in that design? The assumption is that every person they put into the pot, into the study, is genetically identical, culturally identical, and responds the same way physiologically. And that's simply not true. Now, I'm not a statistician, so you can argue with me about that, but I just showed you the genetic data, and I can show you dietary intake data, which would suggest that. So if you actually look at what they're doing, is they're putting in more noise and not increasing the signal because they're putting in more genetic heterogeneity and more environmental heterogeneity. So I used to work for Bill Slicker, and he has in his office this sign here, which I liked, and so now I use it. We're doing the same experiment over and over, and we're expecting different results. And it's obvious that that experimental design was very useful in the early part of the 2000s and all the way up to the time when we can actually start measuring genetic variability and lifestyle variability. I would claim now that we have to move on. Now, I still live by that data. I want to make sure I stay away from the toxins you guys tell me about. I want to eat well. I exercise. We live about twice as long from 1850 to 2010. On average, our human species lives about twice as long. And it was because we did these types of studies. So that was very useful. But now we can move to the next level. So that's what we're going to talk about for the rest of this. So what my career has been about is looking at experimental models and asking questions about how do you design them to look at gene-environment interactions. So in the 1990s, I did a lot of work with mice, and I published some studies with that. And we didn't call it EQTLs then, but we did overlaps of gene expression with quantitative trait loci. With NCTR, we have a very good team that we're still working on the use of stem cells and nutrients, and we'll be publishing some papers. But I want to focus today on the idea about how do you do a human study, or how do how am I trying to do a human study which takes this variability into account. So what we always have in the back of our mind is that it's a gene-environment interaction. We heard today how you tried to do a GWAS study in which you look at the statistical main effect of the genes, because you didn't measure in most GWAS studies the environment. And there are genes which are going to be regulated because a human's a human. I grant that. It's going to happen. So we will find those genes in a GWAS study. And then if you're an environmentalist or a nutritionist or a toxicologist, you look at the environmental exposure and forget about the genetic makeup. But what we actually have to look at is the statistical main effect of the interaction term. And if you don't measure the genotype and you don't measure the environment, you can never get that term from the study. And therefore, I would claim that the models you build without that data are not going to be accurately representing what goes on in the biological system. So I call this slide my Darwin slide. And I have to remind my genetic colleagues who in publications have said that nutrition doesn't matter, that environment doesn't matter. We can find everything by looking at the genes. Well, if you believe that, then you don't believe in Darwinian evolution, in which our species adapted to environments and were modified or selected, selective pressure. It wasn't all genetic drift as we came out of Africa. We selected on things like malaria. We also selected on nutrients because of the availability in the environment. So in order to understand biology then and responses, we have to measure both genotype and environment. And the way we do this now is that we want to look at patients or subjects over time. We look at homeostatic measures, which I will tell you soon are very hard to look at because there's so much variation in the human population. We look at lifestyle and dietary assessments. We want to do genomics and, when possible, and appropriate epigenomics. But most importantly, we want to look at how the change in a biomarker occurs if you have a medical or a lifestyle intervention. And that change relative to your genetic background is what we would really like to look at, and that will give us the most information about describing the system. The reason we have to look at the delta and not just a homeostatic measure is because we've known for many, many years there's a huge variation in human homeostatic measures. So we believe that if you perturb homeostasis, then you can measure the robustness of the response, and that will give us an indication of your current health status, but also could predict your future health status. And you can think of gestational diabetes as one model, right? A woman gets pregnant and there's a stressor, which is the pregnancy. A woman who has that stress or has gestational diabetes has a 5-fold increased risk of diabetes within 5 to 10 years of that delivery. So that's an example of that. So we, in fact, want to use oral glucose tolerance as one of the stress that you would get. And depending upon the response that you would have over time and at the final level of glucose, you can put people into a normal glucose tolerant, impaired, or diabetic. But instead of just measuring glucose or insulin, we would measure all or as many serum metabolites, immune function, oxidative damage, a complete systems analysis to assess that health. So that's one of the approaches that we're looking at. We think that there can be, of course, nutritional challenges. There could be activity challenges like exercise, cognitive challenges. We also would like to do over-the-counter drug challenges because, as an example, acetaminophen causes liver problems in a certain subset of individuals. Perhaps if we had the right markers, we can identify that. I think the Hamner Foundation put out a study in mice using acetaminophen toxicity and early response. So the idea is out in the literature. The most important thing that we want to stress, though, is that you need the deep genotyping and deep phenotyping. That means complete analysis. But we have no reference population. And that's a very important point. We normally study Europeans or European descendants, and then we assume that the responses are going to be the same throughout the world. And we know genetic variation around the world is so great that that response won't work. So, of course, the way we do this is in the 1950s and 60s, the nutritional or epidemiologists would look at what diet you had and what clinical markers, high fat, cholesterol, and that would give us some health information about what to eat. our geneticist friends came along and says, we don't need lifestyle at all. We'll just look at your genes, and those genes will predict how you respond. And then, of course, every technology, the proteomic people, the metabolomic people, the epigenomic people, all of those individuals, camps, said, oh, we have the magic technology. We don't believe that. What we believe is you measure your lifestyle. That lifestyle interacts with your genes to create an a priori, which then will modify the expression of your RNA, your proteins, and your metabolites. And that pattern of all of your factors in many different measurement tools will give us a metabolic group that we can put you in. And we think you will be in a metabolic group regardless of how heterogeneous we heard about human genotypes and phenotypes are, because there is not an infinite range of variation within a human in genetics or in lifestyle. So that's the conceptual basis of what we do. The idea then is not to put people into a case control, but rather just get them into a room and characterize them as deeply as you possibly can. And then you ask, how are they alike or different relative to the response that you had? So I don't put, just by looking at someone, say, you're a BMI of 27, therefore you're overweight and you're in the overweight group, because the person at BMI 27 can be perfectly healthy. I can't look at somebody and know. So I'm going to let the data drive the analysis. So you can do this in a relatively small set of individuals. Then you go back. If you have enough money, you do the whole analysis in a separate population. And then you ask, what's the probability that I can put people into the same response group? So my view is that the new analysis or the new way of doing work is not the power of the experiment, but this probability to put us into the right group. So I'm going to show you that that works. So there's a second design. I showed you the homeostatic response. Some nutrients and toxins are slow acting, so you have to expose people for longer. So we worked with the USDA in Arkansas, and we worked in a summer day camp in which we wanted to look at vitamin levels in response to a whole food intervention. So we looked at metabolites in the blood at the end of the intervention, and 1 month after they went home. We did genomics and DNA methylation, and because Eric, if he's still in the room, we're not going to talk about methylation because we did it in white blood cells, and I know that doesn't work. We were setting up technologies, too. We did 24-hour intakes, activity measures using accelerometers and skin tone. To my knowledge, this is the most completely characterized children that I've seen in a study. So if we were to report the group average, this is what we would get for S-adenosylmethionine, S-adenosyl homocysteine. So here's the group. But in reality, the data that we got for the individuals was this. And then we did a K-cluster analysis, and we found two groups of children. 10 of them had a high SAM-SAW ratio, and 10 had a low SAM-SAW ratio. And this was essentially empirically derived. We didn't expect this when we started the experiment. Now that we got it, we can ask a lot of questions about why those two groups were like that. The other aspect that we did is we looked at other metabolites in the blood. The SAM-SAW was in red blood cells. So for each metabolite we measured, and we only measured 8 others, you could report the group average and say, here's what the kids had in that study. But the data actually looks like this. So what we did is we sorted the data from high to low. And then we said for each metabolite, if I have a published reference value, then we'll call that one standard deviation above the mean will be individuals with a high response. And there's one example of a data point and where it would go. One standard deviation below the mean would be a low level, and then everybody who has levels in between. So each bar would represent one individual and their tertile or reference value relative to the reference data. So we did that with all of the metabolites, and here's the pattern that we have. So in this particular case, here's the high SAM-SAW, and I hope you can see without me drawing the lines, but there's our patterns here. So the high SAM-SAW group here and the low SAM-SAW group. And then there are patterns of metabolites. So if you have high SAM-SAW, you have high vitamin A and vitamin D usually, and you have low vitamin E, thiamine, and pyridoxal, and then the lines are a little bit off. And then if you have low SAM-SAW, the reverse pattern occurs. So here we're looking at an individual. Each row is an individual. Each column is the metabolite. And therefore we can look at the individual response, but we can also group the individuals into like metabolic responses. Now, of course, you can go down to the individual, and we want to, but at least this gives us the first example where we can now look at both the individual and a grouping based on their response, in this particular case, to metabolite levels. We also then could explain why the kids were in different clusters by the diets that they had. In general, the kids in cluster 1 ate more, and they had higher levels of many of the nutrients. That's just 24-hour intake. And then we measured the metabolite levels as groups in the high and the low, and we also found metabolic differences between them. So in the last 2 minutes, I'm running a little bit late, but what we next wanted to do is try to look at the genetic profile of the high SAM-SAW group. Could we explain the difference in the responses based on genetics? Instead of looking at a single gene and a SNP, which, come on, you got to give us the fact that a single gene is not going to really explain a complex phenotype. Or looking at GWAS, in which we know we don't have a lot of kids. We only had 100 in the study and 61 with all 3 data points. We said that let's define a micronutrient system, subsystem, of these micronutrients, and there were 428 genes in the GeneGo pathway analysis which metabolize, interact, or are regulated by these genes. Then we said how many of the SNPs in those genes are on the Illumina array, and there are 5,295. But these are the ones we actually measured, so we then asked another question of, let's just look at those that we measured, and there are 3,870. And I'll show you data on those two. We also tried 26 1-carbon genes. So here's the pattern for the all metabolites, the 5,280. These black bars are the high SAM-SAW group, and these red bars are the low SAM-SAW group. These are frequency of greater than 0.5 SNPs, highly reduced because we only had at each site 0, 1, 2 for homozygote, heterozygote, homozygote. However, I hope you can see these patterns, which are that there's a distinct difference between the kids in the 2 different groups. 2 kids apparently are misclassified, and so there are 184 SNPs and 81 genes which are statistically significant between the groups. So this is an in-group analysis of only those genes involved in the pathway. So these 2 kids, actually the way we did the experiment is each blood sample was independent, but we ran the study in 2009 and 2010. So if a child came back in 2010 who was in 2009, the DNA would be taken separately, and so would the metabolites. So it turns out that this child that has the pattern that should be up here is actually child number 1. This is where they were located in 2009. In 2010, they came back, and they were nutritionally, metabolically in the separate group, but their DNA was the same. So this shows the GWAS, and the second one is the second child here, who also had a different metabolic pattern. So this says to the GWAS people, you got to measure the metabolites, but also the diet. I'll show you that data in a minute to explain those 2 kids. We also are playing with the data in the good sense, and we said take the duplicates out and only look at the highest or most significant SNP in each one of these 81 genes, and we still see this pattern. So child number 1 in 2009 on average looked like the high SAM-SAW group in terms of dietary intake, so did the second child. When they came back to us, they reported essentially that their diets were less good or different. And that for both children, they then looked like the low SAM-SAW group. And when we did their metabolite levels, the same occurred, measuring quantitatively the values in the children showed that in 2009, they were more like the high SAMSA group, and in 2010, they were more like the low SAMSA group. Something changed in their environment. And these are the genes which are statistically significant between the two groups. So many transporter genes, the genes involved in one-carbon metabolism. Some mitochondrial genes, in fact, they're the carnitine transporters. Of course, many genes involved in intermediary metabolism as well as in regulation. This is a complex phenotype. We're measuring 10 separate metabolites. Why would we expect a single gene in the one-carbon pathway or a transporter gene to reflect this great change that we're seeing in the metabolite levels? So what we're saying is that this is a new approach for looking at our studies. Now, we have limitations, particularly for this study. Our genetic spectrum was very small. These are African Americans in the Delta region of Arkansas. So we need to expand the genetic spectrum. The food and cultural diversity was confined as well because of where we studied. And we didn't really study well the physiology of the individual. Now we're asking the FDA's IRB to look at 1,000 proteins in the samples that are left in each child. Many of those will be inflammatory markers and other markers of health, so we hope that we can do that. But the advantages are that we can look both at the population level data, we can identify individual data, we can group individuals into like responses. And we think that this model can be applied to drugs, can be applied to nutrients, and actually can be applied to small doses of things that we think are dangerous. And in addition, as I showed you, we now have brand new nutrition questions to ask. Do we actually need all high levels of vitamins? If salmon SAW is a good marker of health, some kids had low levels of some of the vitamins when they had good SAM-SAW ratios. So now we're going to ask new nutritional questions. So in summary, we want the deep analysis of the response to acute or long-term interventions. We want whole genome analysis. We have to compare across many populations. We want to quantify before data reduction, not classify first and then look at the differences. And for the time being, this approach, which Sidney Brenner in a conference called the middle out approach, instead of looking at all genes or one gene, you look at some system in between, like a Google map. You're not at the earth level, you're not at the street level, you're somewhere at about a 10,000 foot level. We think might have a very strong benefit to look at genomic data. So this is the team that we had in the Delta. I'm very proud that 25 of the 28 people on my team helped us work with the people in the Delta, our collaborators. And before I left NCTR, Bill Slicker, unfortunately, was not there, so I didn't get a chance to say goodbye to him. This study would not have happened if I did not have his support, so I thank you, Bill, for that. We put it in 2008 saying we would not have case controls, and that was not well received. But I think now that we've got the data, I think it justifies this approach that we're trying to look at. And also, we had very huge support from our friends in the Delta who were participating in the study. So finally, I really think this is great data, but this is what we can do to stay healthy. Thank you.
I think we only have time for one question, so Cheryl, you were first.
Okay, thank you very much. That was very, very interesting. You may have said it and I may have missed it. Oh, sorry, Cheryl Walker, Institute of Biosciences and Technology. This type of disruption in one-carbon metabolism could have very profound effects on the epigenetic patterns. Homocysteine is a potent inhibitor and so forth. Do you have the opportunity to go in and see if these SAM-SAW ratios actually correlate with global differences that then could account for global changes in gene expression?
Well, Eric mentioned it earlier. I don't know, but Eric mentioned it. We did DNA methylation in whole blood cells, but we didn't divide up the cells into different types. So part of what we were doing with this study is setting up the systems to study. So we wanted to do methylation, but I don't think we can use it. Also, you have to ask the question of whether white blood cells is the best surrogate marker for us. Could very well be, but we don't have
For a global disruption of one-carbon metabolism, it should work just fine.
Right. If you did it over 90 days, and then we could be able to see it. Yes, I agree with you. But you'd have to have the cells turn over and look before and after. Sorry for being so long.
That's right. Thank you. It's very interesting.